And now we're going to get to the meat of the issue, which is how do you then use it to do spatial phylogenetics? So I hope we've been able to explain as much as we could in the time that's come. But what I want to switch gears to now is actually looking at some of these metrics that we're using. Hope people can see my screen now. So we're going to examine two parts of biodiversity, and whether you're talking about species diversity or phylogenetic diversity, it's conventionally partitioned into three levels. What we call alpha diversity is how much biodiversity, whatever your metric is, how much biodiversity you have at one location, like a particular grid cell. Beta diversity is how much difference there is between locations. So how much difference is there between this place and that place? And together, those two components make up what's called gamma diversity, which is the total diversity of a site. And it can be measured with species counts, and it has the same distinction. But for our purposes today, we're measuring using the branch lengths in the way we've discussed. So in this section of the workshop, we're going to talk about alpha-phylodiversity. How do we talk about how much diversity is in each place? And in the next section, we're going to get to beta phylodiversity, which is how much change is there between places. Both of them are very interesting, but you got to kind of discuss them one at a time. And this gets back to the data input questions that we had earlier. The two basic sorts of information, which are two different digital files that go into the analysis. One is a spatial data set that has the names of the tips of the tree and location information on them. The other is a file that's in a format that's called Nexus, which is neither here nor there, but it's basically a text file, but it's a particular format that has the alignment of the DNA data in it and the tree that's been inferred from it. It can also just have the tree if you've pulled the tree out of some kind of a database of trees. But you have a tree file and a spatial data set. And think of it this way, those data input sets are the gamma diversity. You put those in for everything that's in your study site. So if my study site is California, then I'm going to have an overarching phylogenetic tree for California, and I'm going to have an overarching spatial data set for California. I have to have that even to calculate my alpha diversity. I have to know what the whole region is. So sometimes that's called the species pool, but more generally, we'll just call it the lineage pool. The taxa lineages that are anywhere in California are in the tree, and all their spatial data, as good as we can represent it, is in the spatial data set. And now what our particular problem is, and we're going to look at alpha diversity of a place like one grid cell, is we're asking how much of that total gamma diversity is in that place. So the whole phylogenetic tree is 100%, and I'm going to look at what percent of the tree is in each place. So it's very important to keep this global local distinction in your mind. So the two metrics we're going to use, two of which I introduced before, but I'll mention again. The phylogenetic diversity is basically the proportion of the global phylogenetic tree that's present in a particular place. It could be as much as 100% if every organism is in that space, but it's usually less. And then phylogenetic endemism is that PD weighted by how common the branches of the tree are. And I'm going to show you with it an example of what I mean by that. And there are two derived measures that we're going to use a lot that I want to introduce first, which are ratios. Relative phylogenetic diversity is the ratio of PD on the original tree, whether it's a chronogram or a cladogram or a phylogram, to another topology, which is where all the branches are equal, like the cladogram. I'll show you an example of what that means. And the relative phylogenetic endemism is the same kind of ratio. It's PE on the original tree versus PE on this comparison tree. And we're going to get a statistical significance out of it by randomizing the occurrences of the taxa across that whole gamma area, the whole study site. So this just illustrates what I mean by PD and RPD. Here's the original PD tree over here. This is a cartoon tree that I'm going to use in several slides here to introduce these concepts. So this is a cartoon that just has 5 taxa in it for simplicity to view it. And these are the branch links, in this case represented as a phylogram, so inferred amount of change. We're going to compare the branch links here to the branch links here, where this is basically the cladogram, where we have added or constrained the tree such that all the branch links are equal. So I'm going to compare that branch length to that branch length. And the reason I'm going to do that is because I want to locate places on the map where there are unusually long branches or unusually short branches. And I'm going to generate a null hypothesis for that in a way I'll explain in a minute. The same way is this tree that you measure PE on. I'm going to explain in more detail in a minute, but it's basically this same topology where the branches have been shrunk down by their extent of their range. And I'm going to measure PD on this tree and compare it with the range-weighted comparison tree. So I'm going to explain how these two are generated, but I just want you to see at first that these are derived measures that are comparing the original heterogeneous branch lengths with one where they're forced to be the same. And if you find an extreme value of RPD or RPE, it means that there's a lot of long branches. If it's long, if it's a significantly short RPD, it means there's a lot of short branches in an area. Okay, so here's the same cartoon tree. Let's refresh in our minds how we do phylogenetic diversity, which you've already explained, but just so you can see it in this real example, there's our 5 global members of this tree. And let's say in a particular location, we only have 3 of them present, and we want to know what the PD is in that location. The units that we usually measure the branch lengths in is proportion of the total length of the tree, rather than number of nucleotide changes or number of years. And the reason for that is to make it easily comparable between trees. So this particular branch on the tree right here contains 20% of the length of the tree. This branch down here contains 25% of the total length of the tree. And then there's some shorter branches in here that contain less of it. And if I wanted to say, what's the PD of this particular selection of 3 terminals, I add up the branches that are solid here, and I leave out the branches that are dotted here because they're not included in that sample. And when I add those up, I get a PD of 0.9, which means 90% of the tree is present in a location where those 3 things are present. So that's kind of a high PD. And it's because I artificially picked the long branches to add in there. If I pick the shortest branches and say, well, what if it's those 3 that are in a place, then I would add together the branch lengths that are solid blue here, and I'd leave out the dotted ones. And the PD of that selection is only 55%. So only 55% of the tree length is present in that site. And then here's an intermediate case. So what I would do is measure, I have 3. There are only certain ways you could have combinations of PD with those 3. But if you built up a random model where you just said, well, I'm going to take 3 over and over again, and what PD do I get? That's pretty much how we get the kind of null model of it. And then we look for outliers, which I'll explain in a sec. So PE is exactly the same as PD, except we're going to adjust for the range size. So this is the same exact cartoon tree and branch lengths, but the red numbers here are added, and that refers to how many grid cells that particular terminal is present in anywhere on the map. So this one over here is highly endemic. It's only present in 1 grid cell. This one here is pretty endemic. It's only present in 2. But these terminals here are more common. They're found in either 4 grid cells or 5 grid cells. Now, what I'm going to do is I'm going to divide each branch length by its range size. So I'm going to divide 0.15 by 5. I'm going to divide 0.05 by 2, and so forth. And I'm going to get a topology that looks somewhat like this. So the branches that are long here have been shrunken down, and the branch that was quite endemic has stayed the same size because it's been divided by 1. And just to blow up that restricted part of the tree so you can see it, this is not to scale, but if you blew that up, you'd see that these two, which were sister, and in this case, this branch was 3 times longer than that branch. Once we do the range weighting, because this one has a smaller range, these are nearly the same length. So the length going up to this one is proportionally longer than the length than it is in this tree. So it's a sort of a 3-to-1 ratio here and nearly 50-50 here. So you see, you're preserving the branch length in the branches that are less widespread. And therefore, when you measure PD on this range-weighted tree, it's still a PD measure, but it's a special one because it's emphasizing the branches that have a smaller range. And that's really important both for academic purposes and for these conservation purposes we're going to come up to. Okay, so RPD, to go back to that, how would we calculate it? Here's our comparison tree over here again, as I showed on the second slide of this section. I'm going to take the PD I get on my original tree, which is the same as I showed before, and I'm going to contrast it with the PD that you get with that same selection of taxa in the comparison tree. And in this case where I had added in the long branches, you see I get a relative phylogenetic diversity of 1.2. So the concentration of 3 branches of this selection is more than I would expect if the branch lengths were all equal. So I'd say, oh, here's a place where there's lots of long branches. When I take this selection that had the short branches, I see a RPD here, 0.73, that's much lower than 1. And so therefore you would say, well, there's some short branches there. And then you have intermediate values of RPD depending on which 3 you had picked. So if you find significantly high RPD, it means there's a lot of long branches in that location. And if you have significantly low RPD, it means there's a lot of short branches there, which may not seem like a lot, but it means a lot, as we'll explain. Especially the relative phylogenetic endemism. Here is a review of what the topologies look like. There was that nice animation that we had in the first section, but here's the original phylogram in A, and then a range-weighted version of it. This is for the California flora. And then here is the comparison tree, the denominator of the 2 ratios. And then here's the range-weighted version of it. So when we do RPD, we're looking at this versus this, A versus C. And we're looking at RPE, we're looking at B versus D. And particularly when we get to RPE, you might start to see the significance of it, because when we have a significantly high RPE, it means we have a lot of long and range-restricted branches. So that's getting us into maybe a center where these long branches are, which, as it turns out, is going to be what we call paleo-endemism. So here's a quick example of the alpha-phylogenetic diversity patterns from the California example that was published in 2017. This is open source paper, and you can look at the details there online. The upper 2 maps here are, on the left, the taxon richness, and on the right, the weighted endemism of the taxa. So that's a non-phylogenetic measure at the top. And then on the bottom 2, it's the phylogenetic diversity on the left and the phylogenetic endemism on the right. And you see there is apparent correlation between richness and PD, so the 2 left ones, and there's some correlation, visually at least, between the 2 patterns on the right, weighted endemism and phylogenetic endemism. So, the expectation is that those should be pretty similar. And here's the motivation behind that. So if you thought about it, if the particular taxa that live together are a random selection of all taxa, and if there's no cause happening where taxa don't care who they live with and there's no environmental differences, then you would expect, if you pick 7 taxa from the tree multiple times, you'd expect there to be a high correlation under that null model of ecology. That would be expected to be quite similar between taxon richness and PD. And so what we're looking for are outliers. We're looking for places where the relationships of the taxa that live together are not as would be predicted under a neutral model, but are actually interesting. So in other words, in many cases, organisms do care which ones they live with. They're not living in random assemblages. They're actually living in assemblages that are influenced by environmental and biotic causes. And so to test the statistical significance, what we do is you take the occurrences off the map electronically. Computer does it. You take the occurrences off the map and you put them back on again randomly with the 2 constraints listed there, where the richness of each grid cell remains constant. So the number of terminal taxa in the grid cell is constant, and the range size of each of the terminal taxa is constant. But otherwise, they're shuffled on the map. And so if the original diversity or richness of a site was 7, its richness is always going to be 7, but they'll be randomly selected from the tree. And if you do that 1,000 times and measure PD and PE and so forth on all of those replicates, you'll start to get something that looks like a bell-shaped curve, which is the random expectation of what PD should be if kind of nothing's happening, which scientists call the null hypothesis. And then we're going to look at outliers. We're going to look for the extremes of that distribution and see if our real data are not as predicted by the random. So that's the basis for these kind of maps, which were also published in that paper, and you can read more about them. The top one is the PD randomization. The red places on the map of California to the right there, upper right. The red areas are where the organisms that live together are more closely related to each other than is expected by chance. And arid areas tend to be that way around the globe. And it's because the kind of lineages that can live in random areas are constrained by their traits and their ecology. And then interestingly, the blue areas, including some areas in the northern foothills in the Central Valley, are where the lineages that live together are significantly distantly related to each other, which may illustrate another ecological principle of competitive exclusion. That's what you would expect if the traits of the organisms are kind of pushing against each other and leading to a case where you have one representative of this lineage and one representative of that lineage. That is a concept that goes back to Darwin. So you can get ecological information out of these PD patterns. And then at the bottom is this RPD randomization, which remember is a slightly different question. This is whether or not there's a lot of long branches or short branches in a grid cell. And this probably relates more to biogeographic and evolutionary processes than strictly ecological ones. But it's the places with concentration of long branches that could very well be a climatic refugium of some sort, where lineages have accumulated over time. And so you get a lot of long branches in there. So maybe coast redwood forest would be an example of a place where you see high RPD. And then short branches in this case are places where there's likely to have been recent diversification. Going back to that human analogy that I used before, and this is what we use with the press sometimes kind of half jokingly, is that the blue centers there are kind of like old people's homes where there's a lot of longer branches with a lot more experience. And then the red areas are the nurseries where there's or preschools where there's a concentration of younger lineages, in one case humans, in another case organisms. So we might be able to use these methods to point us to places where there's active and recent diversification. occurring. So we get some sense of biogeography and evolution from just these simple patterns. And then the most interesting, particularly if you're interested in the conservation aspects, is the RPE, which I explained before. So that's PE treated the same way as RPD, where you're doing PE on the original tree versus PE on this comparison tree. And we're trying to find our centers of endemism, which are very important both for academic biogeographic questions, like why do we have organisms that are rare clustered together? And also, especially for conservation, because these are some of our targets for importance for conservation if they are a region that has a statistically high concentration of taxa that have small ranges. So what we do, it's complicated and I don't want to explain it all, but just to make sure you understand how it works, I'll have a diagram in a second, but we're using this RPE ratio in a two-step process. We're first determining if a place is a center of significantly high phylogenetic endemism. So you have to have significantly high PE after this randomization happens that I explained. So I randomize occurrences 1,000 times and I look at the tail of that distribution and compare my actual PE with that tail. So that's what we call a one-tailed test because you're only comparing with the high tail. And if the grid cell passes that, then we look at RPE for that grid cell and we parse it into one of these categories that I'm going to show in a second. And that has to do with whether that's a two-tailed test, and that has to do with whether or not RPE is significantly high or low. When it's a center of endemism that's significantly low in RPE, then that is one of these centers that's been called a center of neo-endemism, meaning that there's a lot of young lineages in there that are range restricted. And vice versa, when it's significantly high in RPE, the interpretation is that's a center of paleoendemism. That's these older long branch taxa that may have accumulated together because of some biogeographic process or climate change. And then there is mini grid cells that are not dominated by either of those two types of endemism. So we call those mixed endemism, and we separate that as a category on the map. So you get a map, and before I show you the map, I just want to show you how it works. This is the same cartoon tree, and this is the same tree I showed you before, but now we're just focused on this rare short branch that was here and the rare long branch that was here, and we're looking at what happens with them. Here's the range-weighted original tree, then here's the comparison tree that we use in the denominator of RPE for these guys, and all the branches have been adjusted to be of equal length. And we notice that when we range weight this tree and this tree, then we find that the long branch in the original tree is shorter in the comparison tree than it was in the original tree. So it's going to be influencing that RPE ratio to be big. The short branch is longer in the range-weighted tree than it was in the original tree, which is going to push us to the side of neo-endemism for an area, especially if there's other confirming taxa like that. So the way Canopy works is that it picks out branches. It can find both paleoendemism and neo-endemism. If you just did significance of PE only, then you would only focus on the long branches. And for the reasons we've talked about a little bit before, we want to know both the rare short branches and the long branches. We're not going to ignore either of those categories. So you get a map like this of California from that same paper. And the places that are blue here are the centers of paleoendemism. And the places that are red are the centers of neoendemism. And the purple is where there's elements of both present. And there's lots of interesting things you can infer from this. But one thing that was noted is that the centers of endemism are definitely concentrated statistically on the dry end of the scale. So dry habitats are a harbor for endemism. which are a little bit counterintuitive, but we can talk more about it. It's interesting, and I think has a good explanation. And then just to finish up this section, let's just say that it's been applied to all of North America. We showed you this briefly. There's a wonderful distinction in RPD between the eastern US and the western US, which clearly shows that there's older branches and an older flora in the eastern US than there is in the western US. We've also looked at Chile and many other parts of the world. And, you know, for example, if you know Chile at all, the desert side of it is just like California is, has concentrations of short branches, where the wetter temperate parts of it, even rainforest part down here, has long branches co-occurring together. And then there was this example of the butterfly plant distinction that we've been using as our emblematic illustration all along. And this is an open source paper too, if you would like to read more about how we compared the two. They're actually quite different. We thought they should be the same or very similar anyway because of the pollination relationship, but it turns out they're quite distinct. This distinct east-west distinction that we saw in the plants is not followed with the butterflies. With the butterflies, the long branches are all down through Mexico, and up in the northwestern US and up into Canada, sort of drier areas are where the short branches are, indicating potentially where there's diversification happening. So it was interesting in the fact that the patterns didn't match very well. Here's canopy for the angiosperms on the left and the butterflies on the right, and they're quite different patterns. The western US is, and barely northwestern Mexico, and southwestern Canada is where the endemism is for the butterflies. Whereas for the vascular plants, as you can see on the left, it's you know, concentrated in Mexico, parts of the southeastern US, and a little bit in places in California. I mentioned this in the chat already, and I put the link in there. So if you look at the chat, you can directly link to this webpage. But our colleague Sean Laffin in Australia writes this software and is brilliant. And it's a wonderful software for analyzing biodiversity data of any kind, whether it's phylogenetic or just species-based data. And it's free and downloadable, and it has sample data files, so you can see what the data files look like for input and so forth. So with a little bit of practice, you can actually implement these quantitative biodiversity analyses with or without a phylogenetic tree. So that concludes this part, and we're going to now have a discussion section, answering questions, and follow it with a little break. And then after the break, we're going to talk about two kind of final topics that come out of these basic alpha diversity measures.